Hello, I'm Zach Harrington.
I'm Steve Tracy.
And I'm Lucas Harrington. We're going to be talking about the diversity in soil invertebrates at two different sites of the Nature Center. So the question being asked is, which area of the University of Mount Union's Nature Center would have a greater diversity of soil invertebrates? The two different sites we'll be looking at is an old growth site and a new growth site. The difference between the two is the new growth site has recently been disturbed when it was cleared out for farmland. Alright, so a little introduction. The purpose of the experiment was to observe and record the invertebrate diversity at both growth sites at the Mount Union Nature Center. So with all this talk about diversity, why is it so important? First, it is necessary to an ecosystem because it helps drive the gene pool and can further evolution. And secondly, it can help improve survival of an ecosystem against any sort of disturbance. Finally, it takes a large community of organisms with different traits to help an ecosystem grow and evolve. So, and our hypothesis was that there will be a higher diversity in the old growth site. So, as you can imagine, there have been many different studies done on this topic, and the first one I want to talk about is the one that I reviewed. It is from Stevens in 2007, and they evaluated soil from a pesticide-free. They evaluated soil from a pesticide-free and a pesticide-treated cornfield, and the goal of this experiment was to determine if there was a higher diversity index for invertebrates in the pesticide-free cornfield compared to that of the pesticide-treated cornfield. And they went about this by taking soil samples randomly 7 times over 3 years and extracting the invertebrates using a Burley's funnel. And the results from this experiment, after calculating their diversity indexes, that they were able to see that there was a higher diversity for the pesticide-free cornfield, and we'll talk about the significance of this a little bit later. Another experiment was conducted by Russell Beth in 2002, and he tested invertebrate diversity from disturbance regimes in forest streams. His experiment questioned invertebrate diversity from areas of disturbance, and he gathered data from 25 different areas of disturbance found in forest streams. And upon his research, he concluded that the more disturbance there is in an environment, that there will be less diversity.
And the last experiment that we looked at was by Wickers and Schimmel in 2001. That was conducted in central Sweden. And what this study looked at was how the effect of forest fires could affect soil invertebrates in that specific forest. So what they did is they actually burned a specific part of land, and they got a piece of that soil the day before, the day after, and then 60 days after. And what they actually found is that obviously after the forest fire there was less invertebrates, but deeper within the soil the invertebrates had a higher survival rate, and 60 days later they found out that the soil invertebrates actually began to arise again and survive.
So now we want to talk about the different growth sites. And the first would be the old growth site. The old growth site was an undisturbed area of the forest. And as you can see from these pictures, there were many fallen trees, and most of these trees were a lot thicker in size compared to that of the trees you would find fallen in the new growth area. It was a very filled environment, so there was things falling all over everywhere. A lot more trees present than there was in the new growth. And after collecting leaf samples, we found the dominant trees in this area to be the American beech, the sugar maple, and the cucumber magnolia. The new growth site, as you can see, has less trees, but also has less fallen trees. And this could be that was recently disturbed when the forest was cleared out for farmland, and it is now just starting to grow back to how it used to be. And also, after collecting leaf samples, we concluded that the dominant trees were the black cherry, the red and sugar maples. So the materials we used for this experiment, the first
list is ethanol. We had 4 1-gallon plastic bags, a Hurley's funnel, 2 screens, 2 PVC pipes, a lamp, 2 collecting jars, a dissecting microscope, a petri dish, and a pipette. The first part of this lab, we collected leaf litter and leaf samples and placed them in the plastic bags. And after that, we placed the samples in a PVC pipe that contained a screen and a vial of ethanol. The alcohol was used to extract the invertebrates, and this is known as negative taxa when they move away from the light. This was done over the week span between the labs. The next week, the containers were filled with the arthropods, and we dissected them and placed them in a petri dish, where they were identified with the microscope and used John Myers quick key to soil-dwelling invertebrates to identify them by their order. After this was done, we calculated the Shannon-Weiner diversity indices.
So now onto our results. Here you can see all the orders of invertebrates that we're able to find. I'll spare you guys by not listing them all off. So you see here in NI the amount of actual invertebrate orders that we found. And to use the Shannon-Weiner indice or formula, we're looking for H prime, which is the diversity index, and H max, which is the total amount of diversity that's possible. So the way we get that is, so we have the NI, the amount that we have, and we divide that by the total number of vertebrates that we found, which in this case was 35, excuse me. So that brings us to our PI. The PI was then multiplied by log 10, and then that value was then multiplied by the initial PI. And then we use these values in this fourth column to actually find our H prime and H max. We'll show you here. So the actual equation for H prime is up here. It's the negative sum of that last column, the PI log of PI. And after we added them all together and then took the negative of that, we got 0.793. So then we moved on to the H max equation, which is log sub 10 of the number of orders in our total set, which we had 7. And this gave us a total of 0.845. And then we take the difference between H max and H prime to find the diversity. And the smaller the number, the closer the two numbers are, the higher the diversity. So here we had a total of 0.052, which is a very low number and shows a very high diversity in the old growth site. So here in the new growth site, we went about the same way. Here again, you have the order. Again, I'll spare you and not read them all off. We have in NI the total number of each order that we had, and we went through the same process to get the PI log of PI. And we're looking for the same H prime and H max. So when we went through, we got the H prime of 0.619, and we got an H max of 0.954. And the difference between the two was 0.355, which is greater than the old growth site. So that will bring us into, was our hypothesis supported? In this case, it was. The old growth site had a smaller difference between the two, which shows a greater diversity between the two sites.
And then Stevenson's experiment connects with this experiment because the pesticide-free cornfield is kind of like the old growth site, whereas it's been undisturbed and it has the higher diversity.
Death's experiment also supported our hypothesis because his results showed that the more disturbance there was in an environment, the less diversity there would be, and that's how the new growth site had a smaller amount of diversity because it had been disturbed recently.
And in my study with Wickers and Schimmel, where they cleared out the land from a forest fire, very comparable to the actual new growth site that we had that was cleared out of farmland. And we were seeing that after 60 days where invertebrates started to rise again and start to live, that's the same thing that happened with the new growth site here in Mount Union.
We concluded that the independent variable was the two sites, the new and the old site, and the dependent variable was the order of invertebrates that were found. and that by looking at past research and our own study, the old growth site has a higher invertebrate diversity due to the lack of disturbance in the environment.
References. Thank you for listening.